So welcome. Welcome to today's educational webinar hosted by the American Brain Coalition and the American College of Neuropsychopharmacology. I'm Mark Resnick, and I'm the immediate past chair of the American Brain Coalition and a fellow of the American College of Neuropsychopharmacology. I'm a professor at the University of Illinois College of Medicine, professor of neuroscience and psychiatry, and I am a neuroscientist. So in fact, some of the science that we do is centered on biomarkers, but I'm not going to talk about that because I'm just a moderator. Okay, so the term biomarker is widespread and in general use, and those of you who aren't scientists have all seen it at some point in something you've read or heard. But regardless of the definition, biomarkers hold great promise to parse disease etiology and transform diagnoses, and aid in the development of treatments for disorders affecting brain health. From enabling earlier or more quantifiable diagnosis to supporting personalized treatment approaches, biomarkers have the potential to provide critical insights into a wide range of neurological and psychiatric ailments. And as research advances, they also hold promise for accelerating drug development, improving clinical trials, and helping clinicians better predict outcomes of the treatments of treatment response. So this is really a crackerjack panel we have today, and it's an outstanding group of experts who are going to talk about the different types of biomarkers, their significance in research and clinical care, and recent scientific advancements in the future of biomarker discovery across brain health. So first, we're going to hear from Steve Hyman. And Steve is the Distinguished Service Professor and Harold McPike Professor of Stem Cell and Regenerative Biology at Harvard University and a core institute member of the Broad Institute, which is a cooperative effort between Harvard and MIT. Dr. Hyman is also the former director of the National Institute on Mental Health and the former provost of Harvard. He was a keynote speaker at a recent European conference on biomarker-driven precision medicine in psychiatry, and I was there and thought he'd be an ideal person to anchor our panel. So, Steve.
Thank you very much for that nice introduction, and I will get my slides up. Okay, Mark, is that visible?
That is visible to me.
Okay, well then that probably works for everyone then. Okay, so I am going to give remarks that kind of, as a first speaker especially, contextualize biomarkers, but I'm going to focus above all on biomarkers in psychiatry. These are my outside interests. Let me just say, however, that the teaching slides, that is not the 2 or 3 data slides at the end, because I didn't remember to get permission from everybody to share those, but all of these teaching slides are available as a PDF through ABC. Okay, why does psychiatry need stratification biomarkers? Well, here's an example. It's actually a slide that's 15 years old from the FDA, and it shows prior to this publication, so of course ketamine came later, in the squares and triangles, which simply distinguish trials by the size of their power, what happens when you average the total response, the drug placebo difference, for every antidepressant that was approved by FDA up to that point. And what you can see when you average all of these trials, you get something like a 2-point improvement on the Hamilton rating scale, which is very far from significant. This kind of result actually led some people to say that antidepressants are no better than placebos. But in fact, of course, if you look at person-level data, instead of averaging the whole trial, you would see, and that data is available, though we don't have time for it, that about 24% of people in these trials actually have an extraordinarily good response, more than 16-point improvement on the Hamilton. Another 50% respond modestly, and you can't really tell, if you're being honest, whether it's drug, placebo, or simply passage of time. So the drugs have been approved, but clearly these are very expensive clinical trials. I should add that every single approved antidepressant has had at least one failed trial where placebo was at least as strong a signal as the drug. And what one needs are biomarkers not to diagnose major depression, which was the incoming diagnosis, because major depression is a syndrome. What you need are biomarkers that will allow you to subdivide this population, that is to stratify them, is the tool of art. So for those of you who have not seen this, there was a FDA NIH working group that reported 10 years ago, BEST as an acronym for something I don't recall, but it defines biomarkers as a characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or response to an exposure or an intervention, including therapeutic interventions. Now, that gives you the sense that a biomarker can be any biological correlation. In fact, useful biomarkers for brain health need to be much more than that. They become useful when they are the result of a rigorous demonstration that demonstrates their robustness, their replication, and their utility in defined contexts of use, which I'm going to come to next. But just to make this point, if you want to use a biomarker for treatment development or even clinically, it has to be relatively bulletproof. I mean, you can't send off a patient's LDL cholesterol assay and get back something that is random. You're going to make clinical decisions, whether initially on drug approval, but day to day in the clinic, on the fact that this assay has been demonstrated beyond doubt to have implications for LDL cholesterol level, and that has been associated with cardiovascular events and mortality, and the assay is reliable, and that's really what matters. And then biomarkers are not, again, one thing, not a correlate. They exist in contexts of use. So psychiatry really does not need biomarkers for DSM-like syndromes. There's no time, but there's an old tragedy of the dexamethasone suppression test, where the idea was it was supposed to be a diagnostic biomarker for major depression. The problem is that major depression doesn't exist in nature. It is a conventional human collection of symptoms and signs. It is a syndrome. What biomarkers are needed for are for stratification. The other thing to be said is that biomarkers are not specific. These are not specific diagnostic tests. These are simply guides in a context of use. So a widely used biomarker, neurofilament light chain, was used to approve an antisense oligonucleotide for SOD1 ALS, but it's also positive in Alzheimer's disease, and it's positive to a degree in schizophrenia. So the fact that it works across diseases is not a problem because you interpret it in that context. You're not using it to decide whether somebody has ALS. You know that somebody has ALS for many other tragic reasons. You are using NFL then to see whether they're getting worse or getting better. Psychiatry needs to be able to stratify these syndromes. Depression, schizophrenia, bipolar disorder are very real, of course, with respect to the distress and the disability, but they are not real as being natural kinds. These are the definitions created by committees based on 1960s and 70s science. And what we need is not to make a diagnosis of schizophrenia, but rather to identify the unknown number of mechanisms within that heterogeneous syndrome. And that's going to take us many, many years of research. But we have not yet actually, as a field in psychiatry, turned our attention to doing this in a good way. And I will come back to that. We also need tracking or staging biomarkers. Disease progression matters. So everybody probably knows that the monoclonals against amyloid beta are not likely to be helpful once the disease has progressed past a certain point, and that can be determined by biomarkers. And then there are treatment biomarkers. And again, in psychiatry, what's really important with high placebo rates, also, by the way, true in Parkinson's disease, is to have objective markers of treatment response that let you distinguish response from placebo and to understand why a drug might or might not work. Now, this may be obvious, but why do we need biomarkers in psychiatry? Well, as in all of medicine, diverse mechanisms can give rise to clinically indistinguishable conditions that require different treatments. In the old days, a syndrome like cough, we didn't have before, I guess, chest x-rays and thermometers and other things we take for granted. We couldn't necessarily tell the difference between a cough due to a bacteria, a virus, inflammation, pollution, or cancer. Clearly, we're well beyond that for crude syndromes like that. But psychiatric diagnoses are, quote, descriptive. That is, they rely on self-report, on clinical observation, and they are entirely surface level descriptions. As a result, psychiatric diagnoses, everything in the DSM, except the diagnosis of narcolepsy, which does have a number in it, is blind to underlying mechanisms. In fact, disorders such as major depression and schizophrenia are caused by an unknown number of mechanisms, and it's really important that we recognize that there are not going to be distinct subtypes, each one due to a single mechanism. The mechanisms we already know overlap, and we know that because we've looked even at the level of genetics that people with any of these diagnoses have overlapping polygenic risks. So that if you have major depression, but you have a high polygenic risk, subthreshold for having schizophrenia, but a high polygenic risk of schizophrenia, you're more likely to develop psychotic symptoms if you have a very, very severe depression. Well, the upshot of this heterogeneity without biomarkers is not just the very unfortunate clinical trial result in the FDA slide I showed you at the outset, it's true in all case control studies, because the cases in psychiatric case control studies, if we have recruited people with major depression or schizophrenia, are heterogeneous. And that means that, of course, they'll need different treatments. But for case control studies, it results in what we know all too well, which is studies that give small, poorly replicable effects. Even back when I was NIMH director in the 1990s, so eons ago, last century, we could see this problem emerging in case control studies using structural MRI. And in fact, 30 years of case control studies in structural MRI has not produced a diagnostically useful distinction. So if somebody comes to the psychiatrist or to any physician with a new onset psychiatric disorder, the only indication for a structural MRI is to rule out an underlying structural neurologic lesion. Now, there are some alternatives being proposed to this, but I think the point is there's nothing wrong with the technology. It works brilliantly for basic cognitive neuroscience, for example. But when you have underlying, actually unrecognized normal variation and superimpose on that syndromes with an unknown number of underlying mechanisms, it's unsurprising that we have not produced something clinically useful. So what do we need? We need biomarkers. If there were orthogonal biomarkers that were applied to patients undergoing structural MRI, I suspect we would have something much more useful right now. For clinical trials, the inability to stratify patients has led to very high, in fact, the highest clinical trials failure rates in medicine, and that despite the fact that there are very few novel mechanisms of treatment being tested. And this has led industry basically to give up on us and to reallocate resources to more scientifically mature areas such as cancer, immunology, metabolism, and of course now neurodegeneration. I think it's important that there are, of course, new psychiatric drugs being developed, but that all of them, including the latest, which is xanomeline, combination called Cobenfy from Bristol Myers, again, was discovered serendipitously by observing humans, so not by basic science. This you can have if you want the PDF. It's just to say that we have to be careful not to imagine that there are one-to-one correspondences between the kind of pathophysiology, the technology of a biomarker, and the treatment. So, for example, for the development of drugs, pharmaceutical companies and biotechs prefer fluid biomarkers such as cerebrospinal fluid or blood because they are looking for molecular mechanisms to be rectified. But in the end, they may very well want to look at imaging biomarkers, again, if we had good ones, or positron emission tomography biomarkers. Treatments for depression, such as deep brain stimulation now, are looking for biomarkers at the level of local circuits using invasive recording. This is just a reminder that you can find biomarkers at any level of complexity in the brain. What matters is the rigorous correspondence with the pathophysiology and the useful connection to the treatment that is being planned. You'll hear about this perhaps from one of the other speakers. This is just to say that biomarkers have revolutionized the diagnosis and treatment of Alzheimer's disease. So prior to there being first fluid CSF-based biomarkers for amyloid and tau, and now for many other analytes, that then became positron emission tomography biomarkers, about 40% of the patients enrolled in early Alzheimer's disease clinical trials, when they came to autopsy later, they were elderly, didn't have any brain amyloid, so they never had Alzheimer's disease. It's just that clinically, dementia experts couldn't say who had amyloid in their brain until they were examined post-mortem. And of course, now with the use of biomarkers, one can develop new treatments, and they can be required for treatment in the clinic. We're not there in psychiatry. Well, I do want to say something about paired samples, and this is a lesson learned from the Alzheimer's disease community. For about 20 years, the Alzheimer's disease community collected paired samples of cerebrospinal fluid and blood, understanding that blood ultimately is going to be the most useful in the clinic, but that CSF is the best reporter we have on processes occurring in the brain parenchyma. It's not a perfect reporter, but it's the best, or you could say the least worst. Proteins in the CSF tend to be at much lower concentrations than in blood. Many analytes, not just proteins, don't equilibrate across the blood-brain barrier. And after all, if you're interested in a brain disease, you want to know what's going on the brain side of the blood-brain barrier. And many analytes have confounding peripheral sources, like cytokines. The largest source of serotonin in the body is the gut, and after that it's platelets, and only third are the raphe nuclei in the brain. And there are confounding sources of many synaptic proteins. You have the peripheral and sympathetic nervous system. And so I think the Alzheimer's community has established very clearly that what you want to, when you're studying and developing a biomarker, is cerebrospinal fluid. Otherwise, you're probably fooling yourself. However, if you have paired samples, you ultimately can have a Rosetta Stone to interpret blood-based biomarkers. And this is what the AD community has produced. Longitudinal collection of paired samples permitted the approval of a blood test now, such as phospho-tau 217. This is not to steal thunder from the neurology community, but just to say they have set a really good example for the discovery of at least fluid and molecular biomarkers in psychiatry that are going to be critical for new treatment development and ultimately probably for prescription. How are we going to get there? Well, and now I'm very close to the end of my talk, but what's important is I've told you that our diagnoses are syndromes. So we don't want biomarkers for depression or schizophrenia. What's worse, however, is that the DSM, the ICD, and frankly, RDoC and HiTOP are not grounded in a way that one can distinguish the boundaries, or for that matter, the thresholds of an illness. And so if you were to do a biomarker study and recruit people with depression or schizophrenia into that study, you would have a group that is both heterogeneous, but also, well, being too broad, also too narrow. That is, they would be sharing, the people with depression would also have symptoms of anxiety. The people with schizophrenia might also earlier in life have had a diagnosis of bipolar disorder. So my recommendation is that we should, in biomarker research, retreat to large-scale spectra to avoid fooling ourselves, assuming that the boundaries that we traditionally use are mirrored in nature. And so these need not be arbitrary. If you look at genetic sharing, I quote this Grotzinger paper in 2025, where you could use symptom covariation, but you end up with these spectrums like schizophrenia bipolar spectrum, where you could then sensibly, for example, look for fluid biomarkers, or if we had a better theory, physiological biomarkers, and so forth. The key is in biomarker research, if these are going to be bulletproof, replicable, useful, we should use well-phenotyped, genome-sequenced, longitudinal recontactable cohorts, because remember, disease stage matters as well. We should recruit them early in disease. This is difficult and costly. And then we should biobank CSF, plasma, frozen nucleated blood cells to make iPSCs later, and any tissue samples, very much like what's being done for the UK Biobank. But because those people were recruited later in life, we don't see a lot of the more severe psychiatric disorders. And I just want to show you quickly, when you can both begin to stratify using CSF biomarkers from the psychosis spectrum. The different colors are different sites at which samples were obtained, but looking at the complement factors that are involved in synapse elimination, we think that's excessive in psychotic disorders. There are outliers with fivefold more complement than others, and maybe that's a subgroup. I'm not claiming that it is here, but we shouldn't be looking for a single average right answer. We should be looking for stratification. And then the other thing here is NPTX2 is a marker of synapse dysfunction and elimination that was first discovered in Alzheimer's research, where downstream of the pathology, there is synapse loss that is probably causal for dementia. And lo and behold, even with a small number of samples, it is different in patients in the schizophrenia bipolar spectrum, both schizophrenia and bipolar. And all I'm saying here is that the signal size, even with a small number, is very large, which gives me the hope that even though these studies to establish biomarkers will be costly and will take a long time, if you have the right biology, the signal size is pretty large, and you can tell the statisticians to relax. So let me end there and unshare and turn it over to my colleagues.
So we're going to hear now, thank you, Steve, from Carol Taylor Birds, at the Program Director in Translational Research at the NINDACE, Institute of Neurological Disorders and Stroke, and she'll talk about biomarkers in diagnosing and treating neurologic disorders. Carol.
Get myself unmuted. And am I showing the presentation mode or the full screen mode?
Presentation, you're fine.
All right, great. So thank you. And I think this is going to hand off pretty smoothly. Let me just move this a little bit. So no disclosures. So I am not going to go through the definition because we just heard a fabulous introduction from Dr. Hyman. And I won't cover these points as well. I think we just covered that also. So a little redundancy here on my part. Just to reminder, these are the different classifications of biomarkers that were mentioned by Dr. Hyman. And the classification was really helpful because it improves the clarity when we're talking about biomarkers and therapeutic development, and making sure that we're on the same page, thinking about how they're being used and what types of evidence are needed to validate them for different use cases. So as Dr. Hyman was talking about, this is another example of the importance of biomarkers for advancing therapeutics and getting to regulatory approval. This is an analysis from the bioindustry organization, and it's just highlighting, one, what we know, just the unfortunate low success rate of biomarker of therapeutics that actually advance to regulatory approval and get to the clinic. But also the point that between the phase 2 and phase 3 clinical trials is a step where there's especially high failure rate. And having those pre-selection biomarkers, those patient stratification biomarkers to try to identify the right patients for the right treatments is really critical for the success and movement for the therapeutics to advance. So I won't go through this in detail either, just the point that, the FDA established the biomarker qualification program back in 2016, 2017. And this was a publication that was multi-stakeholder working group thinking about what kind of evidence was needed to really have the kind of rigorous evaluation of the biomarkers and their readiness for use in clinical trials. Now I'll say that biomarkers don't need to be qualified to be used in regulatory approvals, but it helps, communicate the types of data that are needed to help ensure robustness and reliability of the biomarkers for the different use cases. And we can think of those sort of more simply in terms of 3 questions, right? Why is the biomarker needed? What's the risk benefit if the biomarker is wrong, if it's interpreted incorrectly? So what's the sensitivity, specificity, accuracy, robustness? And then what evidence is needed and available to support that the biomarker can be interpreted? And I think that's a critical point here. How do you interpret the biomarker for the use that it's being implemented in? So I'm in our division of translational research at NINDS. And we usually talk about translation in terms of sort of this linear progression of how we go from basic biology and understanding towards analytical and clinical validation of the biomarker and implementation. But of course, this is really an iterative process because as we get biomarkers that highlight better ways of measuring a pathology or a biological process, that helps inform studies on the basic mechanisms. And as we get biomarkers into clinical implementation, that allows us to ask, investigators to ask new questions about the disease biology and related conditions. And this is just kind of highlighting the same point and thinking about at different stages, what types of questions are we asking or what are investigators asking? I think there are some aspects in the translational and the later clinical implementation aspect, such as the cost effectiveness and the pragmatic questions about when they're used in the clinic or in clinical trials. But there are many biomarkers that might be very useful for research that might not have the same, they might not reach the threshold to reach the clinic for diagnosis or monitoring, but they might still be very important and useful research tools to help characterize the patients better and help do the patient stratification for therapeutic development. So I just want to highlight a couple of examples. So multiple sclerosis is a neuroinflammatory disease, and it's in terms of thinking about biology and biological mechanisms and understanding the disease, it's one of the more advanced ones in some respects, because there were biomarkers that were being used for advancing therapeutic development in multiple sclerosis decades before many other conditions, in part because of the lesions that were detectable on MRIs and could be used to help assess the responsiveness of therapeutics and patients responding to the treatments that could reduce lesion frequency or numbers. But as imaging methods advanced and other methods advanced, the characterization of multiple sclerosis has become more refined, the diagnosis is improved, and the MS community publishes an updated diagnostic criteria periodically as those biomarkers are validated and help improve the diagnosis. So this is more in terms of the clinical workup. I also just want to make the point that the clinical workflow, it's never just the biomarkers, right? Maybe this is obvious, but we always think in terms of the diagnostic criteria and symptoms, as well as the biology, and how are they used together to advance the understanding of the disease, the diagnosis, et cetera. The other example I want to just point out is Parkinson's disease. And I think, to Dr. Hyman's point before, the neurological disorders, while also extremely complex, there's some advantages, or it's a little bit easier, and I'm not going to say that it's easy by any means, but because many of the conditions start with a known pathology that the disease is defined by, that allows investigators to work backwards, moving from the pathological, the known pathology, and trying to understand what drives that pathology to occur. Sort of the other side of that is looking at the genetics. If you've got a disease that's known to be genetically driven, understanding how those genes drive the later biological processes and symptoms, and to characterize the diseases those ways. So it's not easy by any means, but it is a starting point or an anchor point that helps investigators characterize and understand the disease. I think two other quick points on the Parkinson's I think might be of interest, just to remind people as they're thinking about how biomarkers advance research. The pathology was originally characterized in the brain, but as better methods and new methods were identified and developed to characterize and detect synucleinopathies, which are the characteristic, one of the characteristic pathologies within Parkinson's disease, there was the discoveries that these were also present in not just CSF and blood, although blood is still hard to measure for other reasons, but in peripheral nerves, in the skin, in the gut. And those have led to new questions about how the disease originates, and new hypotheses about what different factors cause and contribute to not just developing the disease, but the disease progression and severity in different patients, and helping characterize those patients and how they may advance at different progression rates to that patient stratification aspect again. So I'm going to wrap up quickly, just, sort of the take home messages here, right? So as we heard already, having biomarkers that can be reliably measured is absolutely critical. And I'll say that I think, we don't want to have, wherever we can reduce variability in how we measure the biomarkers will help improve the interpretation of how to interpret the biomarkers within the overall clinical studies, right? We need to make sure that we have those robust, reliable biomarkers. As I said before, they improve clinical trial efficiency, and they help understand biological basis. So just like the neuropsychiatric conditions, the neurological conditions also are multifactorial in terms of etiology, biology, and different biological processes. So being able to stratify patients and have those additional characterizations and understand how those change over time are just as important in the neurological space to really improve patient stratification and disease understanding. And I will stop there. Thank you, and turn it over to Sarah.
Thank you, Carol. And then Sarah Morris is Chief of the Adult Psychopathology and Psychosocial Intervention Development Branch in the NIMH. And she will discuss biomarkers for diagnosis and treatment of psychiatric conditions. Sarah, please.
Hi, everybody. Are you able, oh,
It's not in presentation mode.
how about now?
Yep, there you go.
Are you seeing
Fine. Great.
Okay, great. Okay, so thank you to the organizers for the invitation to participate in this very interesting session. It's interesting to hear the converging themes coming out across different disorder types. So let me jump in. So we've talked a bit about the DSM, and just for anyone who doesn't already know, the DSM is the American Psychiatric Association's Diagnostic and Statistical Manual, which defines standardized criteria for psychiatric diagnoses. The planning for the next version of the DSM is underway, and this is a paragraph from a recent paper written by the future DSM Strategic Committee, which is in charge of developing a roadmap for the next iteration of the DSM. And they acknowledge that the DSM doesn't include biomarkers because there aren't any. Indeed, they say the knowledge base is rapidly evolving and remains decidedly incomplete. So I thought that was a really excellent summary of the state of the science. And they say that biological measures in psychiatry are not yet ready to be deployed clinically. So yes, at this time, there are no biomarkers with adequate sensitivity or specificity for making diagnoses, for predicting prognosis, or predicting treatment response in psychiatry. And this is not for lack of looking. Their researchers have studied everything under the sun to try to find biomarkers for mental health disorders. It has simply proven more difficult and complicated than expected. But this committee, this strategic committee for the DSM, has a subcommittee that did commit to developing a strategy for incorporating biomarkers when they become available. So the work continues. My comments today will focus on some promising approaches to developing biomarkers in mental health disorders. So taking a step back in time a little bit, that largely in response to this lack of progress in identifying biomarkers and new treatments, the NIMH launched the Research Domain Criteria, or RDoC initiative, back in about 2010. RDoC came out of a concern that we've heard already this morning that that psychiatric diagnoses are extremely heterogeneous, and that NIMH-funded research was perhaps arguably too rigidly focused on those diagnostic categories, and that hypotheses were framed around these diagnoses, and then we might be missing something because the research was so focused on existing diagnostic categories. So we might have been missing signals that might be in the data, but missed because of all the heterogeneity within diagnoses. So the goal of RDoC is to support and encourage research that takes new approaches to studying mental disorders outside of the constraints of established diagnostic criteria. It provides a framework for research to look at specific symptoms or neurobehavioral mechanisms within and across disorders and see if we might improve the signal-to-noise ratio by shifting to focus on more homogeneous groups or dimensions. So I'm going to highlight two specific examples of this approach. This approach focuses on a transdiagnostic symptom. So the BSNP team recruited a group of people with psychosis, including diagnoses of bipolar disorder, schizophrenia, schizoaffective disorder, and they used cognitive and EEG measures. Using those measures, they identified 3 different biotypes that have distinct patterns of brain structure and function and heritability. They then went on to replicate those biotypes in an independent group of patients, and now they're currently testing whether one of those biotypes is differentially responsive to the antipsychotic medication clozapine. So this would be a big deal if they did find that, because clozapine is a very effective medication, but also has a rare but very serious side effect. So being able to prospectively identify a subgroup who is likely to respond, or as importantly, a group that is not likely to respond to this medication, would help greatly in making this difficult clinical decision. Another example, now focusing on a within disorder approach. This work takes an RDoC approach to dissecting heterogeneity in ADHD. So many of you know the clinical presentation of ADHD can be highly variable. Trajectories and outcomes vary widely. So some kids with ADHD go on to develop more problems as they get older, and others don't. So this group of investigators asked if they could identify subtypes within the ADHD diagnosis by assessing temperament and cognition. So this figure just shows a portion of the results based on the measures of temperament. They found 3 different clusters, the negative and irritable, surgent or positive, and the mild to normal temperament. Importantly, they find that this clustering is informative about the likelihood that the kids will go on to develop other mental health issues. So you can see in the red, those children had much higher rates of new mental health diagnoses later in life. So this group's ongoing work is focused on seeing if they can better personalize interventions by including measures of temperament, cognition, and other sources of information. And then I did want to take a couple minutes to talk about genomics. Of course, there's a huge research literature on genomic markers for psychiatric illness. There are many interesting findings about gene variants associated with brain development and how cells migrate into place during development. Very interesting findings about the formation of synapses in the brain, and differences that are associated with schizophrenia and autism and major depressive disorder. So genetic studies are giving great clues about disease mechanisms, but there is no blood test or disorder-specific pattern of genome association. This figure from a recent paper, I think, illustrates really nicely the challenge of finding genomic biomarkers in psychiatry. They looked at genetic data from blood samples in red, the data in red, and also in brain tissue samples in blue, across about 800,000 patients with either major depressive disorder, bipolar disorder, or schizophrenia, and found these patterns of overlapping and disease-specific associations. So the overlapping genes were implicated in a diverse range of mechanisms, including calcium signaling and cell division. But I think this is a very nice graphic presentation of the challenges in thinking about identifying biomarkers or targets for drug development. Are we looking for something more general in that overlapping area, or something more disease specific with less overlap? So just to make the point, I think that Dr. Hyman also made already, that individual data types may be informative, but the future may really be in multimodal biomarkers that combine data types, including static features combined with more dynamic features that might relate to changes clinically over time, and combining biological and more behavioral or environmental measures. Along these lines, 2 years ago, the RDoC unit and NIMH launched the Impact MH initiative, which is a funding initiative focused on projects that take data from the clinical record. So using clinician diagnosis and other information in the medical record as a possible source of important signals that could be extracted and then combined with behavioral data, either from computerized tasks or passive data collected through smartphones, to see whether individual level trajectories can be detected or predictions made about hospitalizations or treatment response using that combined data from the medical record and new data collection. So we funded under this initiative 15 grants plus a data coordinating center, and data collection has started. So I hope this quick overview was informative. Of course, there is so much more. I think these talks just give a glimpse of the really exciting science that is ongoing. I hope this gives a sense of the challenges that the field has faced, but also the really promising ongoing research. And I look forward to our discussion.
Great. Which we, thank you, Sarah.
Mhm.
And we now actually have about 15 minutes for our discussion. And the chair's prerogative, I will start with a question that I had for Steve Hyman. And Steve, during your talk, you said there was no need for biomarkers for DSM syndromes. And my question is this, if in the case of depression, if 80% of depression is being treated by non-psychiatrists, then isn't there a need for them to have a better quantification than those two questions they're currently asking?
So the problem, Mark, is that if we had a biomarker for DSM depression, it would not in any way help associate people with treatment. Again, the FDA data says that in a meta-analysis, Stone et al., that only 24% of people with MDD have a good, in their view, a really strong response to any of the approved antidepressants. Of course, this is prior to ketamine, et cetera. I think that primary care doctors use sophisticated biomarkers and medical tests very well when the meaning is well defined. And what we owe them are mechanism-based biomarkers that are predictive of treatment. I would just add, Sarah pointed out at the end the challenges of the overlaps. Psychiatric genetics suffers from the same epistemic blindness. That is, the Psychiatric Genomics Consortium entered people into case control studies of GWAS and RVAS using DSM diagnoses. And we already know from looking back at the data that the same individual could have been at different times in their life in a bipolar study or a schizophrenia study. And sadly, the PGC, to maximize discovery, that is the large numbers of people to find significant variants, did very shallow phenotyping, and there's no biobanking. So all we really know about these people is a DSM diagnosis, which we know is syndromal and largely biologically meaningless, their biological sex and their country of origin. And so unfortunately, we have to go back and redo a lot of this genetics if we're going to be able to use it for biomarkers and for therapeutic development.
Okay, thank you. Now, we have two questions in the Q&A that I'm going to combine to ask as one. And the first one says, don't we already have a potential psychiatric biomarkers that we don't refer to as such? Lithium levels, weight for eating disorders, anti-NDMA antibodies for psychosis. And then the second question, which is closely related, is we need to widen it. If we widen the definition of the term biomarker, for example, in looking at speech and language differences, can we get to a point where we really create a different kind of understanding?
Yeah, can I talk about speech
And
and
those
language?
two people will tell me if I've screwed up their questions. Okay.
Yeah, if I can jump in about speech and language markers, I completely agree. That is such a promising area and super exciting. I don't know if people are aware of the Accelerating Medicine's Partnership in Schizophrenia project. AMP Schizophrenia is a huge undertaking to look at psychosis risk, and they are including speech and language measures in there, and I think it's a really promising area. But to the larger point, I think I agree that the terminology around biomarkers is problematic because in my mind, a biomarker does not have to be a biological measure. It can be cognitive, psychological, observational, et cetera.
Do you mind if I chime in here too? Because I think the FDA has been working on clarifying some of the terminology, and we use the term digital biomarker all the time. But there are also clinical outcome assessments. And so thinking about, how are we assessing function and functional assessments that might be captured digitally, and how those can be really more sensitive, reliable measures potentially, and that can track changes over time at a level that's just not possible to do when you're doing annual or semi-annual clinic visits. So
Are you talking about digital biomarkers collected in various different ways? You know, mood rings, cell phones, etc.
So, one thing that's important, so lithium levels, of course, it's a very good, replicable, simple test. But interestingly, even though that tells you something about the potential therapeutic benefit and the risk of toxicity from lithium, the secular trend is that lithium looks less and less effective over time. And that is because we don't have a biomarker for lithium responsiveness, that is, for an underlying pathological mechanism. And because there are so few therapeutic options, lithium is prescribed more. People try it, right? And when you enlarge the phenotype and you phenotype shallowly, then you'll include a lot of people who were never responsive. So this is just a way of saying context of use really matters. It's not really what gets named a biomarker, but it's really I like this term bulletproof. How good is the measurement, and how do you interpret it in a particular context? And maybe that's a message I think all three of us really deliver.
Okay, so let's expand that a bit. You mentioned we talked about lithium levels, but do we really know how, in terms of thinking about biomarkers, do we really know how lithium exerts its anti-bipolar effects? Is it GSK3B? I don't think so.
No,
Is it, do we really know how an SSRI exerts its
no.
antidepressant effects? So
So,
how does that play into thinking about treatment markers of treatment response?
well, unless Sarah wants to answer,
Go ahead.
no. Fortunately for us, we don't have to answer those hard questions to have useful biomarkers that could be used by a biotech or pharmaceutical company for treatment development, or and even the wonderful biomarkers that Carol showed us for PD and MS, or the biomarkers for AD, we really don't understand the full pathogenic processes. It's just, again, because psychiatry lacks any diagnostic anchors. Nothing, I think a useful term is neurology is a bit closer to having some ground truth with amyloid and tau and alpha-synuclein and so forth. Psychiatry doesn't. So we're very much earlier in these processes, but that again doesn't make it impossible for us to find highly useful, robust correlations that we can at least use to predict responses.
So then a correlative biomarker is really okay, even if we don't understand how it relates to causation.
Better than no biomarker.
Pardon me?
It's better than no biomarker.
Yeah. Okay. All right. We have two questions relating to donation. One addresses essential tremor specifically, but wanting to talk about establishing a biomarker bank. I think a biobank. And then the other one is, what about brain donation? And is that something that is still being pushed as an important tool to help us develop and understand biomarkers?
I started to answer that one in the Q&A, but I'll just say, so NANDS has a nonprofit forum each year, and that is a great place to get started and learn more and connect with other nonprofits and learn from each other. So you can share that link. And I know American Brain Coalition has been involved there. And also, NANDS does have a brain bank that we help fund, and also biobanks to help groups where they don't have established ones already. So those are some areas of support that are really helpful for the biomarker side, and happy to connect more.
Yeah, I mean, the National Institute on Aging has supported at Indiana University a CSF, but a general biobank that has been widely used by major studies in Alzheimer's disease, the ADNI, which is the Alzheimer's Disease Neuroimaging Initiative, and DIAN, which is dominantly inherited Alzheimer's network. And that's where we're putting our psychosis samples as well, because it's really an excellent biobank. And the nice thing about biobanking when it's well done, and that's the case at Indiana University, is it's also gives an opportunity for sharing of samples and data. And that's really critical because there are going to be new ideas, and you really want to be able to both have samples to test them, but also to know how they correlate with your old ideas.
Yeah,
Now I have
and
another
also,
question.
Stephen, how you touched on the importance of the deep phenotyping
Yeah.
in association with those samples to get the maximal benefit from them.
We
Right.
have another question now, and this really relates to stigma and social justice. And it is, how do we develop psychiatric biomarkers? And I would say, this really should also pertain to neurological biomarkers that are not just precise, but equitable and attainable to those with lower resources and a more diverse audience, especially people living with mental illness that are particularly um, under resourced.
Yeah, I can jump in on that a little bit because it does mention the research funding priorities. It is absolutely essential that biomarkers are reliable and generalizable, that we know the properties for which populations they are reliable and valid, and for which populations they're not. No matter what variables are used to define the populations, whether it's race or socioeconomic status or some environmental factors, all of those are super important, and which plays in to the regulatory processes. Those need to be robust so that those biomarkers are used appropriately and are available for appropriate use. In terms of how we do that science in the current funding environment, all I can say is don't give up. Please reach out to a program officer to find a path forward.
Yeah, I mean, I would say historically, NINDS and NIMH have been very supportive, again, largely in years past, of extending, for example, medical genetics to populations that have been ignored. About 90 to 95% of all genome-wide association studies are done in Northern European populations, not because of prejudice, but because the Scandinavian countries have produced these incredible registries which make it simple to do. But that means that specific effort is needed, right, to engage other populations, East Asian, South Asian, African, and so forth.
Okay, a 2-minute warning. The 10 seconds each. What's the one point that you would like Pete to leave people with? Sarah, why don't we start with you?
Oh man, it's complicated. How's that? 2 words. It's
Okay.
complicated, but don't stop trying.
We keep learning, but I'm going to echo it's complicated.
So if I can build on that, because that's really the truth, right? We in psychiatry especially, but it's also true in neurology, we have to do a better job of recognizing the false boundaries we've created among conditions. So BSNP looked at psychosis, but in a subset of psychiatric disorders, it's not clear that it's pathophysiologically different in Alzheimer's disease, for example. The same drugs work, even though there's a black box for the second generation antipsychotic drugs. And I think by creating artificial boundaries, we decrease the likelihood that we're really going to succeed and really going to understand these things. And I do really love the RDoC approach. It doesn't give us advice on these boundaries, right? But it is a way of thinking that should improve things in psychiatry. Otherwise, we'll just dream that we had clues the way the neurologists do, but that won't get us anywhere.
Okay, so we are at the end of our time. I want to thank everyone for joining us today. If we were unable to answer your questions, we'll follow up individually. And to all of you, be well. Thank you.